Hello, my name is Caroline Troy and I'm a junior at Brown University. Thank you for listening to my poster presentation, Setting Oyster Larvae on Existing Riprap in St. Leonard Creek, Maryland. Last summer, I interned with Morgan State University's Patuxent Environmental and Aquatic Research Laboratory, or PEARL, located on Maryland's Patuxent River. My mentor was Dr. Thomas Eide, and for this project, I worked with him, Richard Lacouture, Amber DeMar, and Victoria Agbula. In this experiment, we examined the possibility of restoring oysters to the Chesapeake Bay by directly setting larvae on riprap. Let's get started with some background. The Chesapeake Bay's oyster population is estimated to be less than 1% of what it originally was, mostly due to overharvesting, disease, and habitat loss. But oysters are important to the Chesapeake region both ecologically and economically. They filter the water, provide food and habitat for other species, and oyster harvesting supports the regional economy. While there are many restoration efforts across the bay, more work needs to be done. The main current method of restoration of oysters involves setting oysters on recycled shell on land, then transporting them to large areas in the bay. There can be problems with this approach, however. Spat, which is newly attached oyster larvae, can have high mortality during transport and placement. There's also a lack of recycled shell to set oyster larvae on, and a lack of hard bottom to place the spat on shell. In our project, we explored a way to directly set oysters on riprap in the bay. By setting the larvae directly on riprap, this method not only avoids spat mortality due to transportation, it also utilizes attachment surfaces other than recycled shells. Our approach also facilitates water filtration all along the smaller rivers and creeks as well as along the coast in larger areas of water. In the future, this method could potentially become a new way for waterfront landowners to be involved in oyster restoration on their riprap and make use of hundreds of miles of riprap structures along the coast of the bay. Our experiment took place at 4 sites on riprap along the Patuxent River in the Chesapeake Bay in Jefferson-Patterson State Park, where PEARL is located. At each of the 4 sites, we had a treatment and a control rock. Each treatment rock had 2 etched 10 by 10 centimeter quadrats, and to be counted in the experiment, oyster larvae would have to grow within the bounds of those quadrats. Experimental rocks were anchored in place a minimum of 1 meter below the water surface at mean low or low water. Treatment rocks were surrounded by weighted curtain containment and then exposed to viable oyster larvae. We hypothesized that a granite rock enclosed in a curtain barrier and then exposed to around 400,000 oyster larvae would have a greater number of attached oysters than the unexposed and uncurtained control rocks. After several days, the curtain was removed, and the rocks were monitored over time for oyster survival and growth. 28 days after release, we examined the rocks for young oysters. We found that 3 of the 4 treatment rocks had small oysters growing in the quadrats. All control rocks had no oysters. To determine if our findings were significant, a paired t-test was used, with our null hypothesis that we would find no difference between the control and treatment rocks' number of attached oyster larvae. We failed to reject our null hypothesis, so our t-test results were not statistically significant with the chosen statistic. However, qualitatively, there was a clear difference between the treatment that had relatively good recruitment and control rocks that had no recruitment. It turned out that the physical conditions early last summer were extreme, with very low salinities interfering with oyster set, growth, and survival. Consequently, we considered these results very positive confirmation that our methods would work even better in more typical early summer conditions in Maryland. To learn more about the larger implications of our experiment, we extrapolated from our average treatment rock results to find a rough estimate of the potential number of oysters that could be established if the entire length of the riprap structure was enclosed in curtain and exposed to the same larvae density, using a minimal estimate of the surface area present on the riprap. We estimated that the entire structure could have supported the attachment of around 33,000 oysters. These rough estimates show the great potential for the wider use of this method of oyster settlement in the future. Looking forward, there are several adjustments that could be made to the experimental design as this project is further explored. For example, the effect of salinity levels should be examined, as oysters are known to grow better in higher salinities. 2019 was a lower salinity year, and the experiment took place in a lower salinity part of the bay. In places like Virginia, the settled oysters may thrive more. Also, the curtain design could be modified to allow for greater restriction of water movement, which would allow more larvae to attach. All in all, our positive results indicate the need for further study of this technique, which would one day support the oyster restoration efforts in the Chesapeake Bay. To finish, I would like to thank my co-authors, Morgan State University's Office of Technology Transfer for funding this project, and everyone else at PEARL. I'd also like to thank AFS for the opportunity to present this poster. Thank you.